Good morning, everybody. So today we have with us Pau Carazo from Valencia. Pau had agreed on coming for a seminar already 3 years ago in spring of 2020. And we had to cancel that. And it took us a while to rearrange this talk. But I'm really happy and thankful for Pau being here today. Pau is in Valencia, in the Instituto Cavanilles of Biodiversity and Evolutionary Biology, Universidad de Valencia. And he's professor titular. And he has his lab there since 2025, when he got a Ramón y Cajal. Pau did his PhD in Valencia in 2010. Then he moved to Sydney in 2011 for a postdoc with an Endeavour Award. Mm-hmm. And then he went to Oxford with a Marie Curie. He has been doing early work on beetles and then reptiles. In Sydney, he was still working on reptiles. And then in UK, he moved to Drosophila. And this is mostly the model system that you're using now, though I think you're still working a bit with reptiles. And the main interests of his lab are the study of behavior, function and evolution of behavior, and also sexual selection and sexual conflict, the biotic and abiotic factors that modulate their intensity.
Thank you. Thank you all for being here. Good morning. Thank you, Rob and Marta, for kind invitation. It's a thrill to be here. Yeah, so I'm going to talk to you today about some of our latest research on sexual selection and sexual conflict in what I've called it the title complex environments. We'll talk a little bit about that later. So the overwhelming majority of eukaryotic organisms use one form of other of sex to reproduce, more than 99% of them. And sex is all around us, and this begs the question, why? But a frequently underappreciated but equally important question that derives from this fact is that virtually all sexually reproducing species are subject to sexual selection. And we have come to understand that sexual selection is a fundamental driver of adaptive evolutionary change, responsible for the evolution of a plethora of sounds, forms, behaviors, morphologies, what we call sexually selected traits or secondary sexual traits. And interestingly, we've learned a lot about the nuances and intricacies of sexual selection since Darwin's first postulated about it in The Descent. And one of the things that we are learning more about is about the consequences of sexual selection, not only to understand the evolution of male and female phenotypes, but also to understand population viability and population evolvability. The interesting thing about sexual selection is that it typically gives rise to condition-dependent secondary sexual traits. So if we picture whatever secondary sexual trait, like the paradigmatic example of the peacock, we have, in this case, males with high condition, high generally good genetic endowment that exhibit elaborate sexually selected traits, and the contrary. And if we picture a population, of course, sexual selection is going to act as a sieve, and it acts as a very effective sieve precisely due to this condition dependence, because this allows sexual selection to purge deleterious mutations with that affect condition, and also capture high-quality alleles. And this phenomenon is what we call genic capture, and it allows to, adaptive evolution, and while maintaining enough genetic variability to operate. And a very interesting aspect of sexual selection is that the demographic costs of sexual selection can be considerably lower than those of natural selection. So if you picture a population again, where we've got both sexes and a variety of alleles, and those with more intense color are high quality or are related with high condition, and the drabber ones are vice versa. So natural selection will sieve out deleterious allele and will capture good alleles by differential survival and reproduction of both males and females. But sexual selection is typically, not always, but it's typically stronger in males, which means that most of the selective costs are paid by males, and females, most of the females typically reproduce. And therefore, the impact on population growth rate, the demographic costs of sexual selection can be lower than those of natural selection. And this has led researchers to investigate to what degree can sexual selection effectively increase population viability and evolvability. And particularly during the last few years, we are accumulating very good evidence that this is the case. So this is a famous study by Alison Lumley in Matt Cage's lab. What they did here is basically they worked with seed beetles, and they subjected seed beetles to several generations of experimental evolution under either strong or weak sexual selection. And after a number of generations, they set up a series of generations of inbreeding with the aim of basically unveiling the mutational load. And as you can see, the mutational load is much higher in those populations that had been evolving under weak sexual selection. And more recently, Justin Calley, Debbie Stewart-Fox, and Luke Holman performed a meta-analysis where they combined all the data available from different studies that had done something similar to this. So basically, experimental evolution studies under strong versus weak sexual selection. And they looked at the impact that evolving under strong sexual selection had for different fitness components for both males and females. And they found that, in fact, strong sexual selection increases different measures of fitness in both males and females, but interestingly so, particularly on females. And this, again, has to do with the fact that males pay more the price of sexual selection. That's what we think. But of course, sexual selection, it can be good for population viability and evolvability, but it can also lead to sexual conflict. The same anisogamy that frequently gives rise to stronger sexual selection in males frequently gives rise to divergent sex roles in males and females. And this means basically that males and females have different evolutionary interests, and therefore reproductive parameters cannot be simultaneously optimized for males and females. And sometimes this gives rise to what we call sexually antagonistic coevolution, an arms race between males and females, in which evolution will tend to favor those traits that allow them to push these reproductive parameters towards their own optima. So sexual conflict over when to mate, how much to remate, how much to invest in each mating, how much to invest in the progeny. And sometimes this leads to what we call the reproductive tragedy of the commons, when this type of sexually antagonistic coevolution favors adaptations in males that allow them to be better competitors against the struggle against other males, so they're better at reproducing, which is the driver of sexual selection, but in doing so, they harm those same females they are competing to access. And this is what we call male harm. And male harm is quite widespread in the tree of life. We'll talk a little bit about that a little bit later. And in typical adaptations for male harm range from traumatic insemination to forced mating, sexual harassment, toxic ejaculates. It is worth noting that some of the adaptations for male harm are collateral consequences of males struggling for access to females. And this is the case with forced matings and sexual harassment. In other words, males don't really gain anything by harming females in these cases. It's just a collateral consequences. And some people actually talk of a pleiotropic harm in these cases. In other cases, such as traumatic insemination and toxic ejaculates, and interestingly, these are cases where males are competing for sperm competition, the males do gain by harming females. And typically what they gain by harming females in traumatic inseminations or in toxic ejaculates is basically they decrease female receptivity, so females are less prone to mate with other males. Also, females invest more in current reproduction because their long-term survival is compromised. And these two things are good for males and obviously are bad for females. And so in a recent paper, we collected all the information available on direct and collateral harm adaptations, or at least adaptations that are supposed to be harmful for females. So the balls here denote the number of studies, the number of reports in each of these groups, and the triangles basically denote the fact that there's indirect evidence that this type of adaptations are very common in this type of group. And as you can see, there are interesting trends. We can talk about that later if you want. So vertebrates mostly exhibit collateral harm, whereas insects exhibit both direct and collateral harm. But the main thing we wanted to test with this is there really evidence that this type of adaptations are costly for females? So we ran a meta-analysis, and what you can see here is an ordered plot. So here we're just showing the average fitness of control males and control females, sorry. And control females in this kind of studies are females that are reproducing under low sexual conflict scenario versus females that are reproducing in a high sexual conflict scenario. And this typically means a more male-biased or more equalized sex ratios. And as you can see, there's a strong difference between fitness of control and females under high sexual conflict. Fitness of females in the control treatments is significantly higher. Interestingly, the variation in female reproductive success is lower in control females. So basically, the takeaway message here is, given the evidence we have across the tree of life, yes, male harm adaptations are bad for females. They decrease female fitness. They also increase the variation in female fitness. And this might have interesting consequences for what we expect to happen with female resistance. We can talk about that later if anybody's interested. And so there's good evidence that male harm is indeed harmful for females. There's this nice study by Jean-François Lereliard showing that this can actually lead populations to extinction, or this bring them closer to extinction. And there's a couple of recent papers showing that this type of harm can actually offset those same demographic benefits of sexual selection that we were talking about. And we also show in a very recent study that this type of male harm can also affect the dynamics of adaptation and evolutionary rescue. So the question is, we've got this phenomena that is widespread across the tree of life, but what factors modulate sexual selection and sexual conflict? What factors modulate, what factors explain or push populations or species along this axis between cooperation and competition during reproductive interactions? And this has been the main focus of the research in my group over the last few years. We've looked at different types of factors. We've done some work looking at the potential role of kin selection. I'm not going to talk about this today, but there's some papers here if somebody's interested. We've also looked at other demographic costs, such as aging, demographic factors such as aging or dispersal rate. But here I'm going to focus on our latest research. And our latest research aims to understand the ecology of sexual conflict. And of course, we've long known that ecology is fundamental to understand sexual selection. Here's the Emlen and Oring classic paper. But we know very little about how sexual selection unfolds in what I call here complex environments. And I'm using the term complex environments here, since to Kristin Miller and Eric Svensson review, which basically means that we know that different species or different populations living under different ecological conditions that affect, for example, resource distribution and things like that are going to be fundamental to understand what happens with sexual selection. But any given population within any given species will reproduce and will compete for reproduction under a range, a variation, a fluctuating ecological conditions in both space and time. And we know very little about what happens within this fluctuations, because most of what we know about sexual selection is either conducted in the lab or in the field, but it integrates all this variation. So we don't know actually what happens with sexual selection mechanisms or with sexual conflict with this variation. And this is our main aim. And we think that temperature is a perfect factor to focus on this kind of research for obvious reasons. It is one of the main abiotic factors influencing organism phenotypes, but also it exhibits wide daily and intra and interseasonal variation. So species must have adapted, populations must have adapted to reproduce and to compete for reproduction over this continuum of variation, right? There's also some recent evidence showing that metabolic rate could be a cornerstone, a mechanistic cornerstone to understand the relationship between ecology, life history, and sexual selection. So, for example, some recent work by Göran Arnquist group beautifully shows that in seed beetle species, ecology, resource distribution, determines basal metabolic rate, and this actually is fundamental for the evolution of life history, pace of life, and the economics of mating, and the evolution of sexual conflict, sexual selection dynamics. And of course, temperature is a fundamental determinant of metabolic rate, particularly for ectotherms. So we were excited about trying to understand how might temperature or variation within the adaptive range in temperature affect sexual selection in general. This is work conducted by a former postdoc, I think former also PhD student here at the museum, and current Marie Curie Fellow in Lund, Roberto Garcia Roa, who's, I believe, is sitting right there. So the first thing we wanted to understand is to do a conceptual review. We wanted to understand through what processes might this type of variation affect sexual selection and sexual conflict. And sure enough, there's not much evidence, but there is very convincing evidence that this type of variation can have very direct effects on sexual selection by modulating the opportunity for selection, the covariance between fitness and sexually selected traits, even the heritability of sexually selected or secondary sexual traits. And many indirect effects via effects on reproductive costs and benefits, demography, trade-offs, and correlated abiotic factors. And we also conducted a preliminary meta-analysis. Preliminary simply because there's very little evidence. So this is a meta-analysis on studies that had manipulated temperature fluctuations and looked at how this translated in terms of variation in male and female reproductive success. And the more variable reproductive success, the stronger that sexual selection can be. And so we found a general effect, so that when we had fluctuating temperatures or temperature fluctuations tended to increase the variation in reproductive success in males. And this, interestingly, was not driven by stressful temperatures, but also by this natural, what we call here, natural variation in temperatures. Interestingly, in females, we did find an effect, but this was driven by stressful temperatures so an interesting difference between the sexes. But again, this has to be taken as a preliminary evidence because the evidence is well, we don't have many studies, so hopefully we'll be able to do this again in a few years. And then most of the work that we've done since then has been using Drosophila melanogaster and focusing more on sexual conflict. And Drosophila melanogaster is a fantastic model system for sexual conflict for a variety of reasons. The first of which is that it has an intense history of sexual conflict. And because it's a model organism, it has been amply studied. So we know we have characterized the mechanisms of sexual conflict very well. And in this species, sexual conflict, male harm to females, comes basically through two types of adaptations. We've got pre-copulatory harm, harassment, that basically has to do with male aggression rates and male courtship rates to females. And well, males do what you're seeing here. And this is a low sexual conflict environment for females because here it's just one male and one female. But if we had more males there, you would see the males constantly fighting over the female. And this eventually injures the females, but it also comes with very high opportunity costs for females because in the mating patches, females have less time to forage, less time to oviposit, so it's quite costly for females. And the second avenue of male harm to females in this species, post-copulatory harm, has to do with what has been called toxic ejaculates. So here is as a cartoon, basically a scheme of the reproductive organ of males and females. And if you look at the reproductive organ of males, you've got the testes there. For those of you who are not familiar with Drosophila melanogaster reproductive system, I guess most of you. And then you've got this huge accessory glands here, whose only function is to produce proteins and other substances that are going to accompany the sperm and are going to be transferred to females. And there's been a lot of work on, on this type of substances, and there's a bunch of these substances, seminal fluid proteins, that actually manipulate females. Some of them are necessary for correct reproductions, but a few of them are actually harmful for females. And what they do is they basically reduce female receptivity. So when females receive these substances, they lower their receptivity to other males. They increase their short-term reproduction, and they live shorter lifespans. And in most cases, this basically reduces female fitness at the expense of male fitness, because this makes males better competitors against other males. And what you have here is just the cells that are producing one of the most famous seminal fluid proteins, what is called the sex peptide, within the accessory glands. Okay, so yeah. So I'm going to talk now about the work of Claudia Londoño Nieto. She's an outstanding PhD student that I've been lucky to supervise over the last 4 years. She's almost finishing her PhD. And she's been looking at the phenotypic plasticity of thermoplasticity of sexual conflict, and also at some evolutionary responses. So we've been working mainly with wild flies, so from a population that's near Valencia in the interior. And this is an interesting population because it has to reproduce under a quite broad range of temperatures. So the first thing we did is collect wild flies, and we set up a stock population in the lab, let them acclimate for a couple of generations. And then we collected eggs and let them develop at 24 degrees plus minus 4 degrees. And this is basically a fluctuating temperature regime that mimics what these flies encounter during the reproductively optimum moment of the year. And then we incubated, or Claudia incubated these flies for 48 hours at either 28, 24, or 20 degrees. And then she studied sexual conflict. And the way we do this with Drosophila, as with many other organisms, is basically you set up a high sexual conflict scenario for females. That is one female with 3 males. And males are going to be competing against each other and harassing the female and transferring more seminal fluid proteins to the female. This has all been very well studied in Drosophila, and a low sexual conflict scenario. And the way to measure the degree of male harm is basically just to compare the lifetime reproductive success of females here and here. So how much does fitness decrease, does female fitness decrease when in a high sexual conflict scenario? And this is basically what Claudia did. She looked at the effects of temperature on net male harm, but also the mechanisms, the known mechanisms of male harm in this species, so male harassment and ejaculate effects on females. And this is the main finding. What you've got here is the temperature treatment, 20, 24, and 28 degrees. And there's lifetime reproductive success of females, and when kept in polyandry or in monogamy. So in a high sexual conflict scenario and a low sexual conflict scenario. And the interesting finding here is that the degree to which female fitness is being affected by male harm is plastic, or varies a lot within temperatures. So we've got maximal harm at 24 degrees, which, by the way, is the temperature under which male harm has been studied typically in this species. But we can see that the degree of male harm is reduced at 20 degrees, and it's reduced even further at 28 degrees to the point where we did not find a significant difference. I think there's still harm there. It's just size effect is lower. And the interesting thing too is that the way in which males are harming female, or at least are impacting female fitness, differs between 20 degrees and 28 degrees. So at 20 degrees, female fitness was being decreased mainly through actuarial aging. Females are just living shorter lifespans. Whereas at 28 degrees, it's not so much that they live shorter lifespans, but that their reproductive aging is steeper. So different impacts on different parameters of fitness suggested to us that maybe these different temperatures are affecting different mechanisms of harm. And so we looked at the effects of temperature on pre-copulatory harm, in this case, harassment. And as you can see, at 24 degrees, we get what we expect, which is much higher degrees of sexual harassment in polyandry than in monogamy. The same happens at 28 degrees. The same happens with the levels of aggression. But at 20 degrees, surprisingly, we find no difference between the levels of male harassment between a high and low sexual conflict scenario. And this is indicative of low pre-copulatory harm, because remember, we still have harm happening at 20 degrees, but it's just lower. So maybe this explains why. Maybe what seems to be happening at 20 degrees is that pre-copulatory harm is losing importance, whereas we've got high pre-copulatory harm at warmer temperatures. And another thing that I didn't mention here is that you can see that under monogamy, females are equally happy at the 3 temperatures. So they're well adapted to reproduce at the 3 temperatures. It's not that this has an impact on the reproduction. It all has to do with how temperature affects the levels of male harm. And so we also wanted to see what happened with post-copulatory harm. So we replicated this, sort of replicated an experiment, again, using wild flies that had been reared at 24 degrees, plus minus 4 degrees. And then we incubated males at during either 48 hours or 13 days. And 13 days encompasses the whole spermatogenetic cycle of Drosophila. And then we, at 28, 24, or 20 degrees, and in this case, males were kept either in a high sexual conflict risk scenario. So if you put males with other males, males perceive that there's high competition. And when they mate with females, they're going to court them more, and they're going to transfer more seminal fluids to females. This has been well studied. Well, that's what we expect. So in each case, we had a high and a low sexual conflict scenario. And importantly, then we mated these males that had been kept at these different temperatures in a common garden with females that had been reared at 24 degrees. So basically, any effects that we're going to see on females of mating necessarily have to do with the fact that these males had been kept at different temperatures. And we looked at female receptivity, so how receiving the ejaculate from these males decreased female receptivity. We also looked at survival and at short-term fecundity, which are, like I said, the 3 parameters of female reproduction that are affected by seminal fluid proteins. And this is basically what we found for a treatment duration of 13 days. We see the same general trend for 48 hours, albeit at least a little bit less clear. So as you can see, interestingly, what you've got here is remating latency, so how long it takes for a female to mate with a second male after receiving this first ejaculate. And female receptivity is considerably higher when she mates with males that have been kept at 28 degrees, particularly under a low sexual conflict risk scenario. And this to us is suggesting that maybe seminal fluid proteins are less effective at 28 degrees. And maybe this is why we are seeing so little male harm at 28 degrees. Because remember, at 28 degrees, we have high levels of pre-copulatory harm, but we have low levels of net harm to females. So our guess is maybe it's the seminal fluid proteins that are not functioning well at 28 degrees. And we're looking into this by running proteomic analysis right now. We don't have the results yet. I mean, we are analyzing them. They look good, but it's too early to tell. And then the second part, oops. Sorry, I get too excited when I talk about my research. The second part of Claudia's, I didn't break it. The second part of Claudia's PhD was a long-term experimental evolution study. She collected again flies of the same population that we've been studying, and she kept them over a number of generations under 3 temperature regimes, a 28 degrees average, 24 degrees average, or 20 degrees average, always with a ±4 degree fluctuating regime. So basically what we're doing is we're taking this flies from this population that are well adapted to reproduce and compete over reproduction over this range of 20 and 28, and we're pushing upwards or downwards, and we just want to see how they respond in terms of adaptation. And we had 4 replicates for treatment with an effective population size of 200 individuals. We let each generation flies were allowed more or less 1 week to interact before we collected eggs, and we gave rise to the next generation. We allowed them some spatial structure. This might not look like a lot spatial structure, but typically flies are evolved in a vial. So, you know, there's evidence that basically allowing this structure is important for sexual conflict to operate more similarly as it would do in nature, right? And then I'm going to show the information that we got for 30 generations. We are currently at 60 generations, so we're hoping to replicate the results. So what Claudia did is she then replicated the same experiment that she had done to look at thermal plasticity in the original populations in flies evolved at these different temperatures. And this is basically what we get. This is an index of female lifetime reproductive success. And this is the treatment temperatures for flies evolved at 20, 24, and at 28 degrees. So this is a complex figure, so I'm going to break it down. So basically what we get is a very significant interaction between the effects of the temperature of evolution and the effects of temperature treatment. And if we look at the temperature treatment separately, it's very easy to see what's driving this interaction. So basically, if we look at what's happening with male harm at 20 degrees, you can see that male harm decreases at 28 degrees in flies evolved at 28 degrees. So evidence that, you know, flies are responding to adaptations. Flies are adapting to compete and to harm females at 20 degrees. And it's because harm is lower at 28. At 24 degrees, which is a temperature that was common for all the environments, harm is maximal. And at 28 degrees, we find that harm is has been halved in flies evolving at 20 degrees. So here is where we would have lower levels of harm. And then we looked at the mechanisms that might be responsible for these differences. And here I'm showing aggression rate. Male harassment levels are basically a very similar trend. So what you've got here are the reaction norms for flies evolved at 20 degrees, at 24 degrees, and at 28 degrees. And there's 2 things that we are quite excited about these results. First, as you can see, flies evolved at 20 degrees have evolved to be far less aggressive. And they also court females far less, so they harass females far less than flies evolved at 24 and at 28. And the other thing that is quite exciting to us is that the reaction norm is completely different. So flies evolving at colder temperatures have not only evolved to be nicer to females, if you want, but they are also far less plastic than flies evolved at 24 and at 28. And this is, in conjunction with the results that I already talked about, this is suggestive that at colder temperatures, pre-copulatory mechanisms of harm, for some reason, are either more constrained or are less important for male fitness, and are losing importance with evolution. And then, of course, we wanted to look at the evolution of seminal fluid proteins. So what we did is we performed a series of experiments to look at what's inside those accessory glands of males, of both virgin and mated males, that after evolving at 28 and 20 and 24 degrees. And we performed a multivariate analysis to see whether, what came out. And we found 86 proteins that very nicely grouped our replicates into their respective temperature treatments. And already you can see that 28 degrees treatment is separate from the 20 and 24. And remember, we had evidence suggesting that seminal fluid proteins were not working properly at 28 degrees in the original population. Discriminant analysis, again, very easily distinguishes between the replicates from the 3 different temperature treatments. Again, evidence that they are responding to evolution at the different temperatures. And then we are starting to look, we haven't finished, this is again ongoing, so any feedback is more than welcome, but we're looking very closely at proteins that are overexpressed at 28 versus 20 and 24, and proteins that are overexpressed at 20 versus 28 and 24, etc. And then the first thing that we've done is, are there any seminal fluid proteins there? Just we want to see whether, because of course many of these proteins have to do with sperm storage, they have to do with many other reproductive functions. So we out of the list of proteins that we know have this specific function, seminal fluid proteins, are we finding an evolutionary response? And of the 87 proteins that seem to be responding to temperature, 13 are seminal fluid proteins. And very interestingly, if we look at those that are overexpressed at 28 degrees, of the 11 proteins that are overexpressed at 28, or that we find more at 28 degrees, which would be more precise, than at 20 and 24, 8 of them are seminal fluid proteins. Or another way of looking at this is that 13 of the seminal fluid proteins that are responded are overexpressed differentially at 28 degrees. And this is very nice because we don't find the same thing at either 24 or 20 degrees. So we don't find differential expression of proteins in flies evolved at 24, of seminal fluid proteins in flies evolved at 24 and at 20. And this is suggestive that seminal fluid proteins are evolving differentially at 28 degrees. And the same pattern, you can see the same pattern with mated males. Again, we very easily distinguish between the 3 treatments. And here it's 89 proteins that are responding to evolution. 15 of them are seminal fluid proteins, and of those 6 are responding differentially at 28 degrees. And again, we see no differential response at 24 and at 20 degrees. And now we're looking obviously into the functions of those proteins and trying to find out exactly what those proteins are doing. And we're also looking at the genomics of those, the genotypes of those flies that have been adapting to see whether we can pick up differences between the genes. We also plan to do gene expression, but that's going to take a little bit longer. And so the takeaway message is sexual conflict is very thermally plastic in Drosophila, and it responds quickly to evolution under different temperatures. And very, very interestingly, at least for us, different temperatures are affecting different mechanisms of male harm. So this is highlighting the need to understand these processes if we want to understand how sexual selection and sexual conflict unfolds in nature. And the last result that I'm going to quickly present is work by Michael Butler, master's, very, very good master's students that's been in my lab the last year. And he's been looking at thermal genotype bioenvironment interactions in sexual selection in males. And this is basically looking at an idea that Roberto and I fleshed out in the review into 2021. And we suggested that it is really important to understand the shape of thermal reaction norms with respect to mating success or generally reproductive success in both males and females, actually, if we want to understand how sexual selection is operating. And a very, I think, straightforward to understand this is to compare 2 potential scenarios. So in scenario A, we've got a thermal range at which a species is reproducing. And there's an average temperature here, which in some cases might be optimal if you want. And then we can have this situation. And in this situation, you have genotype A is more plastic, and which allows it to have a higher mating success across a wider range of temperatures. And it's also the genotype that has a higher reproductive mating success in the optimal temperature, right? And the worst genotypes are not only worse in the optimal temperature, but also are less phenotypically plastic. And in this scenario, if we just study sexual selection in the optimum temperature, we are obviously underestimating the strength of the differences in sexual selection. And this other scenario, which I think is more likely, these two, by the way, are perfectly complementary. We expect to find an addition of both these effects. But this one is perhaps more interestingly because obviously we know that there are generally better genotypes than others. But in this case, what you see is alternative strategies with respect to how males might behave across the range of temperatures. So you might have strategies like A strategists that are relatively worse competing at the average temperature, but they're relatively better at the extremes. And this is interesting because this kind of phenomena would maintain or would aid in the maintenance of genetic variation because it's suggestive of alternative reproductive strategies. So to look at this very quickly, again, working with the same population, we derived 40 isolines, so we can have genetic copies of flies within 40 different genotypes, representative from the field. And then we looked at male lifetime reproductive success in a competitive scenario. So what Michael did is we had our focal males, we had several replicates per isoline, and we let them compete against 2 rival males for access to a female over 10 days. And then we switched to a different mating patch where there was different males and different females, and then to a different mating patch with different males and different females. So a pretty comprehensive competitive environment. And overall, the lifetime, we basically looked at how good males were in their competition over reproduction. And we replicated this for each isoline at the 3 different temperatures. So we wanted just to look at the genotype by environment interactions. And this is basically what we got. I was very excited when I saw this graph because normally with genotype, the first thing is that we got a very, very, very clear genotype, significant genotype by environment interactions. But the other thing many of you might be seeing is it seems like there's like only 2 shapes, or basically 2 shapes of normal reaction norms. So a V or an invented V, right? And we tested this statistically, and yeah, that's basically what's going on here. What we're seeing predominantly is either this type of strategy or this type of strategy. So a strategy A, where males are better reproducing at the mean temperature, but relatively worse at the extremes, versus a strategy V, where genotypes are worse competing at this temperature versus these other temperatures. So we're now looking, obviously, into this. We're genotyping these isolines, and we are repeating basically these reaction norms, looking at all the different sexual selection processes, because we really want to know what are these guys doing here better than these guys, and what are these guys doing here that is better than these guys. So maybe, I don't know, maybe male harm is particularly important at 24 degrees, and you've got phenotypes that are particularly harmful to females, but maybe they are better at competing at 20 or 28 because they're better at mate choice, female choice, we don't know. So this is something that we're looking at the moment. And takeaway messages: sexual conflict is bad for females. Male harm is bad for females. Who would have guessed? Sexual conflict is very thermally plastic, far more plastic than we actually suspected it to be. And this I think is quite interesting and opens up very interesting lines of research because it has asymmetric effects on underlying temperature mechanisms. So temperature has asymmetric effects on underlying mechanisms. Cold temperatures seem to be affecting pre-copulatory processes. High temperatures seem to be affecting post-copulatory processes. And male harm and seminal fluid proteins evolve quickly. Also pre-copulatory mechanisms like aggression and harassment. And plasticity, this type of thermal plasticity, can modulate both sexual selection intensity and contribute to maintain genetic variability. So yeah, sexual conflict is bad for females, male harm is bad for females, but this latest, our work suggests that perhaps it's not as bad as we thought, because we had only studied it in a given, very narrow range of temperatures. And of course, many of this work is under construction. So yeah, hopefully I will have more final definite results. And with this, I would just to thank everyone and just my group at the Cavanilles Institute. Thank you.
Thank you very much, Pau, for this interesting talk. It was worth waiting 3 years. Yeah. So we have some questions.
Thank you for the talk. It was really interesting. I was wondering, when you chose those temperatures precisely, I didn't understand very well if you want to draw from that the conclusion that higher temperature will always increase male harm, with something completely specific to that population. I mean, do you think that higher temperatures will provoke a scenario of higher harm, or is just that's very specific?
I don't think that we can expect a general effect. I think the main thing is we chose this temperature simply because we knew this population a little bit, and we knew that we could see them like reproduce in the field under these temperatures. So we thought reproduction was going to be optimal within this range, and then we looked at it and it was optimal. So that's why we looked at it. I don't think you can perhaps some effects might be general, because we know that warm temperatures are generally not good for sperm and things related to sperm. And of course, then males can adapt to higher temperatures. But if you're adapted to a range of temperatures, normally the hottest temperatures might generally be a little bit worse in terms of how sperm is functioning and things associated with sperm. So that might be general. We don't know, because we have very little information of what's going on in other species. But my guess would be that it's going to be very species specific and ecology specific. But at the same time, we might have some general change. So for example, what's going on with at the lower temperatures? It might be constraints simply. It might be that these are ectotherms. At lower temperatures, activity might be constrained, and therefore males might be just simply less able to court and harass females and be aggressive against other males. And in this scenario, they do better investing everything in post-copulatory sexual selection. So this is, we don't know, but.
Okay, thank you. And another question, if I may. For the first paper you showed, there was this meta-analysis of different kinds of sexual coercion and male harm.
Yeah.
Did you consider sexual size dimorphism as a covariate of possible?
Yeah, that's a really, really good question. We considered both sexual size dimorphism as well, we all know that it's a very, very bad index of precopulatory strength of precopulatory sexual selection, but it's what we have. So we looked at that, and we also looked at the intensity of sperm competition. Both came out as parameters that explained the levels of male harm, but only marginally. The problem is that we don't have a lot of evidence. So that is very interesting, because it basically is suggesting that the stronger the sexual selection in a given group, the more chances that these harmful male adaptations are going to actually be harmful for females. Yeah.
Any more questions? So we seem to have a question from someone from home. Can diet, niche partitioning enhance sexual dimorphism in flies? Evolutionary theory claims sexual dimorphism evolved primarily via sexual and fecundity selection, not food competition. But I don't know if the question is finished or if this makes already sense.
Can you read it again, please?
Can diet, niche partition enhance sexual dimorphism in flies?
Can niche partitioning enhance sexual dimorphism in flies? That's an interesting question. I don't know. We know that females are, so sexual size dimorphism in flies, as in many insects, is different to what happens in many vertebrates. So females are typically larger, and that's because there's strong fecundity selection on females. And I don't know, that would be an interesting thing to look at.
I'm sorry, but I have no idea who it was. Do you have any more questions?
Thank you very much for the talk. It was really, really interesting. I have a curiosity, and it might be super stupid because I have no idea about anything in flies. But what happens if you put more than one female?
Yeah, we simplify by putting just one female, but basically, because obviously if you put more than one female, it's difficult to keep track of what's happening with one focal female. But it's basically the same. What's driving the levels of male harm has to do with the sex ratio. So if you have 3 females and 3 males, you would have something very similar to what happens with if you have 1 male and 1 female. And if you put 9 males and 3 females, you will base something very similar to what happens with 3 males and 1 female. Of course, if you consider density, if you control for density, because having 9 and 3 increases density. But otherwise, it's basically what happens. And I didn't say this, but if you study what happens in a mating patch in the wild, because sex ratios are 1:1 for this species, the sex ratios are male biased in the mating patches, which is where females basically go to feed and lay eggs, and that's where the males are waiting for the females, etc. So the 3:1 versus 1:1 are the 2 extremes of what you normally find in nature. So it's an exaggeration if you want, but it's within the biological range.
Thanks for the talk and from my ignorance also about sexual conflict in flies. I was wondering if all these effects of temperature on the sexual conflict in the wild could be avoided by females or males changing their phenology in the mating, because they can maybe move around during the season.
Yeah, that's a really, really good question. That's one of the things that we really want to test. What happens? Because of course, behaviorally they can buffer many of these things. Yeah. So first we want to find out what's going on with these phenotypes, with these isolines, with different strategies, and then what we want to do is how they behave, whether they behave differently at different temperatures, because I think that's critical.
If the females, is bad for the females, maybe they want to move.
And the other thing that we're starting to look at is at the female side of things, right? Because so far we've studied the male side of things because it's the obvious first step, but we also want to study of female resistance. What's happening with females? Because females are not passive players, obviously, in this co-evolutionary play. They respond. So we are very interested in seeing what might be going on with females in terms both of behaviors, like you're saying, how they might buffer these things, and also in terms of physiological maybe responses to seminal fluid proteins or things like that.
And the second question connected to the seminal proteins. I wonder how you will be able to, well, maybe looking at the function, but you know that the geo terms are normally very general about what the proteins are doing really, although in Drosophila there is much more information. But I wonder how you will be able to distinguish if they are just simply generating an environment in which, in a stress environment of the proteins are like a heat shock protein. If this is just that function and nothing else than that these overexpressed proteins are just preserving the function of the sperm, and that's it. And it's not taking care about anything else.
Yeah, that's also a very good question, because we know that sperm does very well at 28 degrees, but these flies that we're adapting to 28 degrees were subject to peaks of 32 degrees. So it could very well be that many of what we're seeing has to do with these types of adaptations. Of course, to begin with, you just have to look at the function. There's nothing else that you can do. The good thing is that Drosophila is a model system, so it's really well annotated. So we know a lot about heat shock proteins. So that would be the first thing. And the other thing, obviously, one would have to do is, I think, knockout experiments to see what's going on.
Thank you.
Okay, do we have any more questions? If not, then we're going to continue this discussion in the kiosco. Thank you again, Pau, for your...
Thank you.